you attach to the endoplasmic reticulum to reload your mitochondria with lipids that are synthesized in the ER, and you upregulate mitochondrial gene expression to rebuild the organelles during recovery sleep.
Yeah. And so basically, right, the longer that mitochondria have been working or turned on, let's say, the more chances there are for them to become dysfunctional, to become damaged. They're generating these reactive oxygen species and these lipid peroxides. That's causing a problem. The longer you let that go without sort of resetting or replacing those mitochondria, the more the cell's going to be in trouble overall. And so that's basically what we're talking about here. The animals are sleep deprived. They're not allowed to sleep. The mitochondria start to become dysfunctional. They look weird. They're aggregating in different parts of the cell, and the cell has to literally digest them and replace them.
That's the idea.
Yeah, interesting. And so we've talked about these lipid peroxides. We've talked about how polyunsaturated fatty acids are more prone to react with reactive oxygen species and produce these nasty chemicals. Talk a little bit more about the endoplasmic reticulum and how the new mitochondria get made. So it sounds like new mitochondria can get made. They kind of dock onto the ER, and this is how they get loaded with like their lipids and the stuff that they're built from.
So this is one of a really important area in modern cell biology that has grown enormously over the past 15 or so years. The study of interorganelle contact sites. So there's numerous such tightly regulated contact sites, including sites that link the mitochondria to the endoplasmic reticulum. We now even have structures in yeast that show the ER mitochondrial contact, and it's in fact a lipid channel. So you can import lipids from the ER into mitochondria. There's a hydrophobic tunnel, and you can pass the lipids through. The reason why you need this, of course, is that many lipid biosynthetic pathways are either exclusively concentrated in the ER or distributed between the ER and the mitochondria. So you have to import precursors from the ER into mitochondria. So this, again, finding the mitochondria attached to the ER suggests that something is going on with the lipids, as we've already inferred from studying the ion channel. Now, mitochondrial fission usually happens at these contact sites between mitochondria and ER. So the sequence that normally happens is the mitochondrion attaches to the ER and then recruits a dynamine-related protein that sort of constricts it and divides the organelle.
Okay, so mitochondria, they can grow in size, they can fuse together, they can split apart, and you can sort of watch and map all of this out happening in the cell, and it tells you something about the health and the state and whether or not these mitochondria have become dysfunctional.
Exactly. And there also seem to be clear relationships between mitochondrial morphology and mitochondrial function. Mm-hmm. So it's widely thought that if you have hyperfused mitochondria, so very large extended branch networks of mitochondria, that allows particularly efficient ATP synthesis, whereas if you have small, rounded, fragmented mitochondria, the efficiency of electron transport and ATP synthesis drops.
Mm-hmm. Got it. So in general, more smaller mitochondria is indication of inefficient energy production, and sort of bigger fused together mitochondria indicates the opposite, it seems.
Yeah.
And so you say in this paper that mitochondrial electron surplus induces sleep.